Well, here we are. We're going to do what I titled Genetics 1. I'm not exactly sure how many genetics sections there will be or lessons, but it's kind of hard to get deep into it without going to, let's say, Genetics 50. So we're going to scratch the surface, peel back the first layer of the onion, and if you are really interested in genetics, it's a fascinating topic, and there are many courses out there that can be had. Okay, so Genetics 1. What did I subtitle it? The Basics and Terminology. So we're going to talk about chromosomes and alleles and a few things, how they interact, and then emphasize terms, because when you do read material, you really got to know the terminology first, don't you? Okay, but it's a fascinating subject because, I mean, look at these two guys here. How can you get any more different than that? I'm going to put it right there, maybe make it even bigger. And they have the same number of chromosomes, those two dogs. Obviously, the Great Dane is on the right. I think that might be called a Harlequin Dane when they're looking like that, if I remember correctly, and the Chihuahua. On the left, two very different kinds of dogs, takes different owners, takes different amount of care for those guys. They represent large differences in costs of ownership, of course. But like I said, same number of chromosomes, same number of bones, same type of digestive tract, same type of reproductive tract. Very interesting. And of course, there are more than 400 breeds of dogs. And we'll talk a little bit about breeds and perhaps maybe disorders, genetic disorders that some of them have. I didn't want to say suffer, but they have these genetic disorders, some breeds. Okay, well, let's start some terminology. And I always try to remember to get these large in case you're on a small screen or something. Okay, well, here we've got the word chromosomes. Well, chromosomes are made up of strands of DNA, right? And here it says each human cell should have 46. They're in 23 pairs, so 23 times 2 is 46. And what happens is you get one of these strands from the dad, which is also called the sire, S-I-R-E. Mom gives you the other one of the pair, and moms are also, in the animal world, I guess, and maybe the genetics world, are called the dams. I'm not swearing now, it's D-A-M. So you have the sire and dam, each contributing one strand of the chromosome number, and so then you get the resumption of the 23 pairs after the egg joins with the sperm, of course. Okay, so then we have genes that are segments of DNA. They contain the instructions. And by the way, back to the chromosomes, I'm going to show you the dog, cat, and horse number here shortly. So genes are the segments on DNA. And then alleles, I always say, are alternate forms of a gene. You know, we often see capital A, little a, lowercase a, and those are two alleles that can fit in what's called the locus, L-O-C-U-S. The locus is the location on the chromosome where the alleles fit. So like down here, I'm on the right now, the allele would fit here, and then another one on this one. So like, let's say, maybe I should get my little, so it could be A on this side, okay, and then on the other side, we might have little a, and those are fitting or sitting on the locus, the location of the alleles. And there I have two alleles available or present. Everybody has, all animals have two copies of the gene at that location. Okay, now let me see. I'm going to take this away. move those letters out of the way and show you the chromosome numbers. And the interesting thing about chromosome numbers, it doesn't mean a lot. It has nothing to do with intelligence or how big the animal is, whatever. So here's cat, 38 chromosomes. Do you remember 46 for the humans? So that's 23 pair. Do you notice all the numbers are even? The number of chromosomes always have to be even. You can split even numbers equally, but you can't if there's odd numbers. So cat, 38 chromosomes. Dog, 78. Quite a lot more, but it's just counting them up. Let's see, where's the horse? 64. And then look at the smallest animal on the list here is a chicken, and it's got the most number of chromosomes. So the number of chromosomes is interesting. In fact, the animals can't crossbreed. It wouldn't work. But the kicker is it has nothing to do with the size of the animal, the intelligence of the animal, anything like that. Okay, moving on to more terms. There's some redundancy here, but sometimes it's good to read two different definitions of the same term because you should know out in the real world different people say it differently. Okay, a gene is the unit of inheritance, right? Directly responsible for a trait, and maybe I should define a trait right now. A trait is an observable or measurable characteristic of the animal. So, for example, coat color is a trait. Mature adult weight is a trait. Okay. Now I could put parameters on those. I didn't say black coat color like I have Newfoundland, so the coat color for those would be black, or so that's observable, right? Or measurable, maybe the adult weight of Onyx, 180 pounds. I could measure him. He weighs 180 pounds. Okay, so that's a parameter there of the trait. Okay, so the allele, I like this definition because it's the alternate form of a gene, I should say. Alternate form. We often see capital A, lowercase a, but maybe there's 10 alternate forms for one locus. Okay, so here it says usually there are 2 alleles for every gene. I'm not sure if that's I'm not sure if that's the right way to say that, but sometimes as many as 3 or 4, and there's sometimes many more than that. So you could have, let's say, 10 possible alleles, but any individual can only have 2, right? Okay, think of the numbers 1 through 10. Those are alleles, and an individual could have 3 and 7. Another individual could have 9 and 1, so forth and so on. Well, it ends up being when the 2 alleles are the same, 1 and 1 or capital A, you could say that's a homozygous state. If they're different, then you say they're heterozygous down here. Okay, so that'd be like, in my example of numbers, 2 and 5. If those were actually alleles that fit into a locus, then that's a heterozygous state. Okay, let's see, trying to get my pointer here. Here's some more genetic terms. And we get into some gene action here a little bit. Dominant alleles. That's when the phenotype of one of the alleles masks the other allele. For example, and let me read what they say. The allele whose phenotype would dominate in a heterozygous state. So if A is dominant and then little a is recessive, then it doesn't matter if the genotype is capital A, or capital A, little a, like in this case, the trait would always be like if there was only an A there. Recessive, that's the allele when the phenotype would be dominated over, would be, let's see, recessive. I'm not sure if I like how would be dominated over. Okay, yeah, the recessive. I guess that's right. So little a, that's the recessive allele. And it's only going to really be expressed when you have the little a. Okay, heterozygote, I should say, is their terminology. 2 alleles are the same. We already said that. Or heterozygous. sorry, I'm saying they're different. Homozygote. I got ahead of myself there. Two alleles of the same gene. Capital A, maybe in my example of numbers, 2 and 2. Okay, here, genotype, the genetic makeup of the individual. So the genotype is, back up here, capital A, capital A. Phenotype is what we can see the outward expression, so what is observable in the animal, like the phenotype of a coat color or the weight. Okay, just to give us another visual here about homozygous states and heterozygous states. My tongue's a little twisted there. Let's look at the far left. Capital B, capital B. The two alleles are the same, and this is the locus. Here's the locus. Remember, locus is the place, the location on the chromosome that has that gene. And remember, alleles are alternate forms of a gene. So this is homozygous, capital B, capital B. Then we have two heterozygous states here, capital B, and then little b, capital B. It doesn't really matter the order because let's say capital B was dominant, those two designations would come out to be the same. The B would be expressed, the capital B. Now here's homozygous, and let's say it's recessive, little b. But you can have B homozygous capital B, and homozygous little b, right? Okay, we're still working on some terms, and we've had some of this before, but we just want to make sure everybody is on the same page. The gonads are the sex organs of the male and the female that make, well, we're going to talk about the gametes, but the testis is the male sex organ, and the ovaries, and there's usually two ovaries and two testes, of course, and those are the gonads. Then the gonads make gametes. Okay, so it's interesting. The gonads make the gametes, and from the testis we get sperm. From the ovaries we get eggs, often called oocytes. A singular egg can also be called an ovum, and then plural of ovum is ova, ova. And then when we do get fertilization, one sperm, that's actually one end, has half the chromosome number, and one egg unite, and the egg also is called what's in a one end state. Those two together give us a zygote, which is that one cell that's now restored the 2N designation. It's got the correct chromosome number for an individual. Okay? And then how did we get those 1N eggs and sperm? Well, that was done by meiosis, I probably should say, and that's called reduction division. You end up with 1N cells, the gametes, after meiosis. Okay, moving on, I want to talk a little bit about cloned animals, and I found this neat little illustration here about what's been cloned and who did it. So that's kind of interesting. Rabbit by the Chinese scientists, the dog, South Korea. I remember when that came. I'm not sure the years for these. Horses, Italian. The French cloned the rat, and then a cat from Texas. It doesn't show Dolly, who was the first cloned sheep back in the late 90s. I remember that. I think it was, we found out about it in 1997 in, can't remember, I think it was, I want to say Scotland. Anyway, you might say those animals are going to be looking very similar. Okay, if this is the donor of the DNA, it looks like, most of the phenotype looks the same. Too bad they didn't show that for the other ones. But the kicker is the animals will not probably turn out to be the same as whoever donated the DNA. I won't get into the actual technique, but here's a little formula that you should remember about genetics. Now remember, phenotype is what you can see. it's the observable trait. Sorry, my tongue's getting tied there. What you can observe, like the coat color or the height at the shoulder, how many hands a horse is. But it's based on the genotype of the animal, right? Like big A, little a. But then there's always environment. So this is quite a neat genetic formula, the genotype of the animal plus the environment. And that's very complex. That can be also the intrauterine environment that the zygote developed in. And that's called maternal environment, the prenatal maternal environment. And then there's also postnatal maternal environment, the nursing, the time that the animal's lactating and nursing. And then, of course, the environment that man or nature provides. Those two work together to show us something. So you can have two animals with the same genotypes, but if they're fed quite differently, then they're going to look different. They're going to be different sizes. So very important to remember that. I don't think I'd ever clone my favorite dog because I know it wouldn't come out to be like the other dog, the original dog. So cloning is kind of neat, but you know, I'm not all that impressed with it. Now I'd like to introduce the term karyotype. Karyotype is like a visual representation of the chromosomes, and they're usually lined up from the largest chromosomes to the smallest. Okay? And it looks like, if I remember right, didn't the horse have 32 pairs? Because it's got 31 down here, but then it's not counting the sex chromosomes. So all these other chromosomes are called autosomes. A-U-T-O-S-O-M-E-S, the autosomes, and then the sex chromosomes up here. But this is a visual representation of the chromosomes, and I've actually done this in a class. So I've got my karyotype, and I've got a dog, and can't remember how many animals I did. You know, you collect actually the white blood cells, grow them, and there's a way to arrest their division, and I can't remember all the details of it, but karyotype, visualization of the chromosomes in a photograph, lined up by size. And I wanted to say another thing about what we see, and we'll get into maybe this a little bit. Coat color is fascinating with horses because there's so many things that are going on. I guess I should have a little lesson about horse coat color because the white gene is very interesting. There's a lot of epistasis going on. Epistasis is where one allele masks, or you could say one locus, whatever's happening at one locus can mask a lot of things. But this just shows you what this is the sire over here, right? And this is the dam, and they mated, and we got this solid color foal. So it's amazing the color combinations that you can get in horses and other animals, of course dogs as well. Now I just want to do a little bit with the Labradors and the three basic colors and kind of show you, give you a little hint of gene action, but we're going to talk about that later. But I want to show you how to interpret these lines for a minute. So let's look at the far left. This is a black Labrador. And a black Labrador has one large B and one large E, and it doesn't matter what the other allele is. So what's that saying is, this could be a large B or a little b here, and this could be a large E or a little e, but you'd still have the black dog. Then we have a chocolate, and the only way a chocolate can appear is when we have little b, and then one E. So that line there shouldn't be there because you have to have two B's. So like, look at, there's a little, let's say a typo. It's got to be BB, little b, and then large E, but it doesn't matter what this is. And then a yellow lab is always little e, and then the other two alleles, the B alleles, it doesn't really matter what it is. Large B, little b, you know, these lines, whenever you see that in a genotype, because this is the genotype, this is the phenotype, right? What you see, the genotype is what the genes, the alleles are really like. But just remember, in this picture, that should not be the case. That line should be gone because it's got to be little b, and then large e and whatever for the chocolate. Okay, so this lesson, Genetics 1, is basically terms and a few basic principles. And then I want to make sure you know the word pedigree. And a pedigree is often a visual representation of the ancestors of an individual. Okay? So let's look at the far left here. We have the individual. So this is a pedigree for this dog, this Labrador Retriever, female. She happens to be black. Well, her sire is up here, and usually the sire by convention is the upper panel, upper area, and her mother, the dam, is down here. So that's her parents. But then we have her grandparents and her great-grandparents. And the sire is always on the top of the pair. So sire, dam, sire, dam. So there's a pedigree for this dog, and it goes back to the parents, grandparents, and great-great-grandparents. Of course, we're still doing like a little introduction, and oftentimes you'll see pictures like these, and they're not photoshopped. That's an actual living cat. It looks like two cats that were merged together, and you know what? That's basically what it is. This is called a chimera. And the definition I have, and I'm just going to say it's two, but two fertilized eggs. Now notice the eggs are fertilized, so you got two fertilized eggs merge into one. And if they hadn't merged, they would have developed into two separate cats. So then here you've got a cat that really looks like a combination of two separate cats, doesn't it? And that's a chimera. And if you would sample from that black skin, the genotype you would find in those skin cells would be different. And let's say the black is on the left in this picture. It's the right side of the cat, but it's the left of the picture. On the left side, the black skin cells would have a different genotype than on the right side of the picture, because there are, in chimeras, genetically distinct cells. So they would have different genotypes. Very, very interesting. Well, I think we're done with our terms of introduction. We might have a few more later, but not many. Here's the list of illustrations I used. Thanks a lot, and see you the next time around.